Hey guys!
Hi everyone!
Welcome back to Pathogens. I'm your number one Jim, Erin.
And I'm your number two Jim, Caroline. And today for episode 4, we have, and we are so graciously welcomed by Dr. Kylene Kehn-Hall, who is a professor at Virginia Tech and also the CESAP director. So she's a big player in the reason that we got this podcast.
Thank you.
She also has a research lab here, and her research focuses on SARS-CoV-2 and encephalitic alpha viruses such as Rift Valley fever and Venezuelan equine encephalitis virus, or VEEV. She studies how these viruses interact with the body and how we can target those interactions to develop treatments and better diagnostics. So, everyone, let's welcome to the pod, Dr. Kylene Kehn-Hall.
Woo!
Mwah!
Everybody, welcome Dr. Kylene Kehn-Hall.
Yay!
Thank you so much for joining us today.
Yes, thank you.
And for our first question today, we would like to ask you, how is being the CESAP director and kind of like what does that entail in your day-to-day life?
Sure. So for those who haven't been on the podcast, maybe I should say what CeZAP is.
Yes.
So CeZAP is the Center for Emerging Zoonotic and Arthropod-borne Pathogens. And this is a center that is really built to bring anyone at Virginia Tech that is doing infectious disease research basically together. So as director, that's my job to really help facilitate this process. And CESAP involves over 100 faculty across Virginia Tech's campuses, across multiple different colleges and departments, as well as students and postdocs.
Kind of on the same, within the same realm, how do you successfully balance your role as the CESAP director along with running a very successful research lab? Because those both have to be very time demanding, I assume.
Absolutely. Absolutely. I would say it takes a lot of careful planning and time management. So, every week it's looking at, okay, what do I need to do for CESAP? I will say first and foremost, I have Sarah Gauger, who's amazing.
Yes.
Yes.
Gosh, we love her.
And so without her as a CESAP administrator, I couldn't do what I do. Mm-hmm. And so she's the one that I go to. Luckily, our offices are very close. And I pop into her office and we kind of lay out, okay, this is what's going on, either this week or this month, and here are our priorities, and let's work to get this done. But I also have really great, faculty within CESAP that help with the various different things that we do. So we have a seminar series. We have seminar series coordinators. We have, obviously, the graduate program, right? We have graduate program directors.
Yeah.
I have an internal and external advisory board. So we have many people that are really involved with helping with all the different moving parts. So careful planning and making sure I have a really good team underneath me to help with all these things.
What do the different internal and external boards do?
So with the internal board, these are members that are at Virginia Tech, and so they really understand kind of the goings on at Virginia Tech. And so we meet with them twice a year to say, these are things we've done, these are kind of our plans, our priorities, and get their input into, how they think we're doing, and also maybe give us ideas of ways to do things better, or maybe new initiatives.
Yeah.
And then the external board, these are members from outside the Virginia Tech community. And so we have representatives from industry, government, academia, and we can, we only meet with them once a year, but it's great to have their external input to say, hey, this is what we're doing, and, tell us from your point of view, what could we be doing better, Usually that's the more, I don't necessarily need to hear, I don't need to hear, like, you're doing great, right? But what could we do better, and what are some innovative ideas that really help us along those lines?
Yeah. Are the external boards, are they mainly like Virginia Tech grads or just kind of people who are interested in CESAP?
It's a combination.
Okay.
Yeah, so we've had some Virginia Tech alum that helped us, and then we have people that we know from maybe industry or government that we've invited to join the external board.
That's awesome.
Yeah.
And then for your weekly meetings with Sarah, how do you guys go about like splitting up the duties? Because you're both very busy individuals.
I think it depends on what's being done. So Sarah's really great, especially if it's something that we've done more than one time. She's gonna take the ball and run with it. Right? All I have to say is like, hey, remember it's that time of year we need to do that, and then she's like, I'm on it.
Got it.
And she's doing it, right? If it's something new, then we'll sit down, we'll brainstorm. We'll bring in Jonathan Geist, who's the associate director of CESAP, get his input, and then we do a little bit more planning up front.
That's really awesome.
This may be a silly question, but for the internal and external advisory boards, is there like if you're on, say I am on the CESAP external board, is it like a certain time commitment that I have to give? Like how often do people rotate in and out of it?
So generally 3 to 4 years for
Okay.
each of them. And we give a little bit of buffer just in case things change in people's life and they need to rotate off in 3 years, or if they could stay for 4, that's great. And it also allowed us at the beginning to have some overlap in people. So for example, we started the center in 2022 and 2020, and I should mention XJ Meng is the founding director of CeZAP.
Yes.
And when it was established, we had our boards established, right? And if they all rotated off at the same time, we would have a fresh new board.
Yeah.
And so we wanted to stagger the rotations a bit, and we've done that with all of our positions where we just kind of staggered people rotating off.
Gotcha.
So since it started in 2020, have you always been involved since the start, or did you kind of come on later?
I have. Luckily, XJ brought me on board at the very beginning. So I actually came to Virginia Tech in 2020.
Oh wow.
Yeah. And so when he reached out to me, actually before I became an official faculty member, and told me about the center, and he wanted me to be a part of it. And so I started as an internal advisory board member in 2020.
Yeah.
Nice.
And then after that, I actually took over as associate director while XJ was serving as the interim director, or the interim institute director for the Life Science Institute.
Yeah. What was that like, kind of establishing the center? I feel like that must have been hectic, kind of.
I would say a little bit, but actually really laid the groundwork. With all the major programs, he did an amazing job kind of setting them up, and so I was really handed, I would say, a well-oiled machine. It was like, hey, and then I could add my own kind of personality to it.
Yeah. That's really great. Yeah.
So you came to Virginia Tech in 2020. Where were you before? Like how did you wind up here?
So I was at George Mason University before that. So I started my lab, I established my lab at George Mason in 2009. And then I came to Virginia Tech in 2020. And George Mason was great. I got recruited to start my program in emerging infectious diseases. And at the time, they were building a biosafety containment lab, like a BSL-3 facility there. And so I got to go there and got to see the facility built and help with bringing up kind of the select agent program. They had to work on the really dangerous viruses, so I worked on. And so I think I had a great time at George Mason, and it just came to a point where I wanted to kind of expand my work and go to an area that had more infectious disease researchers. And Virginia Tech has an amazing wealth of researchers, including virologists. Like I think we're really people underappreciate how many virology researchers we have.
Yes.
It's insane.
Where did you get your PhD at?
George Washington.
George Washington. Okay.
Yes. Right. Where's George Mason at versus George Washington? I feel like I always just
the Georges. Yes. So George Washington is in DC.
Yes.
And George Mason, its main campus is in Fairfax, Virginia.
Okay.
But I was on the what we like to call the graduate campus. That's in Manassas, Virginia.
Okay.
Oh, I never knew that.
It's actually right across from ETCC. Like literally, ETCC is
Oh,
there.
wow.
And their graduate campus is right there.
Nice.
CCC.
Mm-hmm.
Wait, what is ETCC?
Oh, I'm sorry. American
you're
Tissue
good.
Type Culture. It's where everyone gets all their
Oh,
cell
yes.
lines
Okay.
and viruses.
We
or
went to their
awesome.
table. I
We
think we went to
did.
their table.
That which we
Yes,
did.
they were a sponsor at the symposium.
Yes.
Yeah.
That's amazing. Yeah. So is that move really hard to do? Like, I'm assuming you had to move most of your lab stuff with you.
It was difficult, and COVID made it really fun.
Oh,
Yeah,
I bet that was enjoyable. It was great.
It's tough. I think anytime you move a lab, for me, the distance made it a little bit easier.
Yeah.
Because I was actually had a couple of graduate students that stayed behind at George Mason and finished their degree there. I had, faculty at George Mason to help make sure that they were, could keep doing the research.
That's amazing.
It was really great.
Yeah.
And I was able to go back and forth from time to time.
Yeah.
Just kind of check on them. Drive up there.
Yeah.
And also because it was COVID and we were all shut down, we could still do research at George Mason because we were working on SARS-CoV-2, and so they kept doing their research, and then it gave me time to really set up my lab here.
Yeah. Did you start working on SARS-CoV-2 before the pandemic happened, or was it kind of like the pandemic happened, let's look more into this?
Yes, so the pandemic happened,
Okay.
and I think it was just kind of spurred everyone to
Yeah,
action.
really.
Yeah, I hadn't done any coronavirus research before that,
Yeah.
but having access to a BSL-3, you kind of feel
perfect.
that you need to
You
continue
always
in that
inclined,
way.
yeah.
Yeah.
Yeah, and so we definitely were inundated with requests to do SARS-CoV-2
help us.
research. So we took on a lot of that. My group did a great job. And we were extremely busy. We definitely weren't at home with our sourdough starters.
Making like whipped coffee.
I was making
whipped coffee and watching Survivor.
Yeah, that's exactly what I was doing.
Yeah.
And I mean, people were in the lab nonstop. I was
Yeah.
working nonstop, writing grants nonstop.
Yeah.
I bet, yeah.
What was that like to kind of have so many different people relying on you for an especially pressing and publicly understood to an extent pandemic?
Yes, I think it's difficult, right? To have people look at you constantly to kind of explain things.
Yes.
It did get to a point where you know, if you met someone new and they asked you what you do, you're like,
I'm not gonna say
Something.
do I go there?
Yeah.
But you know, I started to learn how to explain it a bit better to people that were non-scientists and try to emphasize the importance of what we were doing and the importance of studying the virus.
Yeah.
So I think that's helpful.
Yeah, for sure.
Yeah.
So we're going to be asking some questions about one of Dr. Kehn-Hall's papers. We're going to link it in the
Description
below. But yeah, do you want to start or you want me to start? You go. Take the reins.
Let's do it.
Okay, so just for like a short little summary, this study basically looked at what happened long after infection with Venezuelan equine encephalitis, or VEEV, which I'm sure you will probably refer to as, because VEEV is much easier to say, which is a mosquito-borne virus that can infect the brain.
So I wanted to mention that the first author on this paper is Maureen van der Heesen, and she is one of the initial cohorts of the IDIJ.
Yes!
And I also should say that the work was funded by the Defense Threat Reduction Agency and is a really interdisciplinary collaboration between my lab, Michelle Theus's lab at Virginia Tech, David G's lab, and then also Eric Johnson's lab.
First of all, what originally motivated studying the long-term effects of VEEV?
Sure. So my work with VEEV for many years, well, first of all, maybe I'll just say that it's a really important pathogen for both humans and for equine, so horses and donkeys. And my lab has been always interested in it from kind of the biodefense type of perspective. So we know that it has formerly been weaponized by both the US and the former Soviet Union.
Okay.
And so we're always worried about it from that perspective. And also it can be lethal.
Yes.
So we always thought of, okay, let's, first and foremost, let's protect people from dying from this really dangerous pathogen.
Yeah.
And so, for years my lab did a lot of work on that, and we're still doing work on that, to try to find either antivirals that target the virus directly or that target host processes in order to save basically patients.
Yeah.
And the more I was studying the virus, I realized that, the mortality for VEEV at least is relatively low compared to some viruses, so around 1%, maybe less. But when you look at the people that are infected, especially those where the virus goes into the central nervous system, so actually gets to the brain, those individuals have really kind of dampened quality of life, right? So they have what's called neurological sequelae. And so they can have long-term emotional issues, they can have long-term memory issues, depending, they might have seizures. And so I started to think about, well, yes, we might be able to prevent people from dying, but what about their quality of life? Can we help from the neurological point of view? And coming to Virginia Tech, I was surrounded by some really great neurobiologists. Some of my really good collaborators with Michelle Theus, who's in our department, in the Department of Biomedical Sciences and Pathobiology. And so with this partnership, we started to ask the question of, okay, what type of damage is actually happening long-term, and then ultimately down the line, how can we prevent it?
Yeah. So when you say that VEEV has been thought of to use as a bioterrorism weapon, how would you go about doing that, as it's a mosquito-borne virus?
So VEEV is extremely easily aerosolized.
Okay.
Oh.
And so just naturally, even in the lab, there's been lab accidents that are not needle sticks where people have, the virus has been aerosolized, maybe by centrifugation. If people, they didn't have respiratory protection on, they would inhale it. And so that's one of the reasons why it is classified as a biosafety level 3 pathogen. We have to wear respiratory protection.
So what does that look like when you go into BSL-3? How are you physically protecting yourself?
Yeah. So in our current lab, we go in and we take off our street clothes and we put on scrubs. And then when we enter the lab, we put on a disposable gown and we put on double gloves. We also have either dedicated lab shoes, so we have like Crocs that we keep in there, or you have to put on shoe covers.
Okay.
And then the most important part is actually your respiratory protection. So we put on a PAPR.
Okay.
And so I think anyone who watched the shows during COVID, right, you saw everyone with the PAPRs on, right? So ours, we have a battery that we wear around our waist, and then it has a hose, and it connects to kind of the headgear. And what happens is that it passes HEPA-filtered air across your face.
Okay.
So anything you're breathing in is HEPA-filtered,
Okay.
and so that you're protected from anything in the air.
Is a HEPA filter just kind of like a really intense filter, I guess, for lack of better terms? Like it filters out everything?
It can filter out a very high percent of particles from the air.
Yeah. Okay. So are VEEV and lab infections, are they less common now that it's known that it can be aerosolized?
Yes, now that we have the proper protection.
Yeah, that's great. Yeah.
How does one go into a BSL? How do I get into a BSL-3? I really want to know what it looks like on the inside.
I mean, you can certainly go on tours. The actual lab, when you go into it, looks very similar to a regular lab. It's more so the PPE, so the personal protective equipment that you wear. It's also the engineering controls. So, for example, the airflow, right? We make sure there's negative airflow, so no air comes out of the lab. So it has to be constantly pulled into the lab.
Yeah.
And then it is the safety protocols you use. So, for example, almost everything has to be done in a biosafety cabinet. So you modify your procedures, so everything's in the biosafety cabinet. When you transport things in the lab, you always have to transport them in like a container, a secondary container. So if you accidentally drop something, it's still contained within the container. There are everything's a SOP, a standard operating protocol, and you have to follow it exactly in terms of even like when you change your gloves. You have to follow it exactly. So you can't take your gloves out of the biosafety cabinet without changing your gloves. Like you can't take your hands out. I think that's one of the hardest things for people is
I feel like
you're
I'm
constantly in and out of the hood, but in BSL-3 you're like, I need to take my gloves now.
So how does that work? Like, do you have new pairs in the biosafety cabinet, or?
So you have a little trash can, like in the biosafety cabinet, so you take off your outer pair, throw it away, with your double gloves.
Yes.
And then you come out and you put on a new pair right away.
Okay.
So I always tell people, you think of your inner gloves as actually your skin.
Your skin, yeah.
So you never touch anything with your skin, right? And your outer gloves, you changing them constantly because you don't want to contaminate yourself or anything around you.
Interesting. Wow. I just want to go in there.
I mean, we could get you a tour.
Oh, thank goodness, thank goodness. Next question is, why is the hippocampus such an important region to study within this paper's context?
Sure. So maybe to give a little more context. So in this paper, we infected mice with our virus, and in this case, we used a live attenuated strain, which is called TCD3, and this allows us to actually do these studies at BSL-2. So it's a strain of the virus that replicates really well in the brain, but the animals can survive infection, and that's key. Like we have to have them survive infection to study
see what happens. Yeah.
Is that why it's BSL-2, because like they survive
exactly.
after? Okay.
Yeah. So the BSL-3 strain will kill the mice.
Yeah.
So we need to be able to study the long-term impacts. And so this study, we infected the mice and we followed them up to 90 days post-infection.
Okay.
And we really honed in on the hippocampus because of its importance for memory. And so we know that survivors of VEEV infection have memory issues or cognitive issues. So we wanted to understand, well, what's happening in the hippocampus? Are there cells that are being infected? Are there cells that are dying? Kind of what's happening long-term as well.
So I was wondering, has there ever been a VEEV case in Virginia or like even the United States as a whole? Because that scares me.
Sure.
I have a bad immune system. I really do.
I mean, it is a worrisome virus.
Yeah.
So it is naturally found in South America, Central America, and there are circulating VEEV in those areas as well as periodic outbreaks. And there have been some cases in Texas of VEEV, so it has kind of crept into the United States. And there are strains kind of related to VEEV that are also found in Florida, like very southern Florida.
What strains?
There's an Everglades virus that is like within the VEEV serological complex.
Does that infect equine and humans, or?
I don't think there's been any clinical human cases that I'm aware of, and so when we think of kind of the VEEV serological complex, we more are worried about be itself, like
Yeah.
encephalitis virus. But some related viruses, there is interest in them because they're just really understudied.
Yeah.
And we don't, it might be that it's impacting humans kind of on rare instances.
Yeah. It's crazy.
I'm on the lookout.
I mean, so not to scare you too much,
Okay.
but there's a related virus, Eastern equine encephalitis
Yes, that's what
virus.
Shannon studies.
Yes.
Yes, okay.
And that is found in the eastern part of the U.S.
Great.
And that is much more deadly.
Okay.
Yay!
Yes, so anywhere from like 30 to upwards of 80% case fatality rate for that virus. And that's with hospitalized patients, so the really severe infections.
Yeah.
And that's also BSL-3.
Woo. Yay. Yay.
Yeah.
Scary world out
That
there.
is a different world.
So if you're going to be worried, I'd be worried about that
I'll
one.
Yeah.
focus on the eastern one first.
Yes.
Once I go to Central America. So.
Good. BSL-3 work. Is that like your favorite type of lab environment to be in? As it seems like a lot of the viruses you work with are mainly BSL-3.
Yeah, I wouldn't say it's like my favorite lab environment because it actually makes research pretty difficult. But I think the viruses that we study that happen to be BSL-3 are really interesting, and they're neglected viruses.
Definitely.
And because they are difficult to study, not a lot of people want to do it.
Yes.
And so we're willing to do it, right? We're willing to take, of course, that personal risk, but we mitigate that through all procedures. But also the time and the effort to really, establish the protocols, do the work, and kind of expand on the knowledge.
For sure.
And so for like Eastern, for example, there's very few labs that work on it, and if you look in the literature, there's just not very much known about it at all because it has to be studied at BSL-3.
Yeah. Is that like or is it less common to have a BSL-3 university, or is it basically every like big research university will have one?
No, every university is not going to have
Okay.
one. So it's selective, I think, and it's going to depend on their research priorities, right? Do they have researchers that study pathogens, whether it's viruses or bacteria that need a BSL-3? And it's an investment in terms of cost for the facility, cost to manage it, regulatory costs, and so they have to make sure that it's actually needed before they invest in it.
So after reading this research paper, kind of what I always like to think is what would the next steps be in this, and it's especially awesome because you're the one who helped publish this paper. So what are the next steps that your lab's taking to like get more information?
So this paper I see is kind of setting the stage for, we know this mouse model is working to model a disease. We know critical pathways that are altered. We know they're still altered at day 90. And for me now the next step is, can we stop these things from happening?
Yes.
So can we look at what we identified and can we intervene? So can we use like host-directed therapeutics to say, let's stop some of this inflammation to reverse the damage, and hopefully we won't have those neurological sequelae.
Yeah.
So that I think is the most important. And then second, because I'm a scientist, right? I really then delve a little more into the details of like, why is this happening?
Yes.
Right?
Yeah.
So why at day 90 after infection do we still see microglial cells that are activated?
Yeah.
And the virus is gone. We don't see it anymore in the hippocampus at least. So why are the neurons still altered? Why are the microglial cells still altered?
Yes.
And so kind of diving into that to understand, so is there, for example, some sort of epigenetic regulation that's left behind?
Okay.
Some signature that's changed and the host isn't able to like kind of revert back to normal state.
Yeah. Is that very unique to VEEV, or do other encephalitic viruses also kind of have that long-term activation?
So the long-term neurological sequelae is not unique to VEEV.
Okay.
So for example, Eastern equine does it, West Nile cases done it. You think about everyone talking about long COVID, right? So I think we're appreciating more as a community that this happens with a lot of viral infections.
Yes.
But I think the mechanisms that lead to that can be different amongst viruses.
Yes.
And so it's really trying to understand that, because some viruses infect different subsets of cells, for example, different parts of the brain. So it is going to be different depending on the viral infection.
Yeah, for sure. One final question. Have you ever heard of Skeeter Syndrome, and would you consider working on it?
Do you know what Skeeter
I don't know
Syndrome
what
is?
Skeeter
So
Syndrome
this is my time to shine.
Okay.
So
great.
last June, this is gonna be a very long-winded story. So last June, well, my whole life I get eaten alive by mosquitoes.
This is like mosquito.
Mosquito.
Okay.
Mosquito syndrome. Okay. So I get eaten alive by mosquitoes, But this past June, I got 2 bites on my ankle, and my ankle swelled up so bad I had a cankle. I couldn't walk on it for like 2 days. Go to the doctor, they're like, I'm gonna sound like I'm dragging my doctor, I'm sorry. They were like, here's an antibiotic. I was like, I don't think this is applicable. And then they were like, just take Benadryl cream, whatever. I go back to work, whatever. They call me and they're like, we think you have something called Skeeter Syndrome. And I was like, oh, okay. So then a whole year passes, and then this past week, I am still dealing with my newest case of Skeeter Syndrome.
It's been bad.
But it's basically where, I guess it's something I just developed over time because it didn't happen to me when I was a kid, but I just have very adverse reactions to a certain protein that the female mosquitoes carry when they bite me. So my leg always is very inflamed after.
Yes.
My bug bites get to be like about this big. It seems
They're like hot and
yes,
rough.
and thank you for asking this question because we
Yeah.
are trying to bring awareness to
we're
Skeeter
trying to
Syndrome.
bring awareness to it and
I'm
see if anyone
asking-
at Virginia Tech would be willing to study it.
and maybe
Yeah.
find something that's not prednisone
Yeah.
and Benadryl cream that could help me.
Because there aren't any papers talking about it. There's a severe lack in literature.
We did a literature review.
Yeah.
So,
Yeah, for sure. I'm sure there's lots of people that react in this way.
Yeah.
I would say lots, but there's got to be other people.
Yeah,
that,
you're not alone.
I'm not alone.
Yeah.
I think my next person that I'm gonna ask is Dr. Waker. If he wants to do some research.
I think he would.
Maybe I can do a postdoc in somebody's lab studying Skeeter
Skeeter
Yeah.
Syndrome.
Yeah, this is my
I go from
long
food
journey.
science to Skeeter
You
Syndrome.
would hate the insectary.
I know, I would.
Yeah.
I would.
Okay.
Amazing.
Amazing. Thank you guys so much for joining us, and thank you, Dr. Kehn-Hall,
Yes,
for
thank
coming
you.
on.
Thanks for
Thank
having
you.
me.
Yeah. We'll see you next time.
Bye.